You got it! Oh my god, I'm sorry crying. I'm just gonna cry. Oh, you're
I was at house, like every weekend after the show. Yeah, obviously, and he's like, I got
If Mike says that, I just can't say shit. That's not my fault. I didn't know what you were doing. You turned around, I was like, oh, yes, and then you saying, oh shit, was it how you're like, oh, yes, you. He was at the maybe you should don't put him on. But you should try to yeah, you definitely shouldn't. That's too sketchy. Especially the fact that you were like, tomorrow? And you asked like, how's my mom? Like, really? I tried to play it off. Like, I do babysat you. Like, I babysat you or some shit. Yeah. No, they don't know really. They're not really your shit over any pussy. Yeah, just say it. What they say. They go, well, what's your name? I go, oh, I think her name was Catherine or something. They go, sick one. I was like, did I? I meant Catherine. I don't know what your idea That's what was what? You should tell. If I learned that, I would have said that. I would have just left. I see this girl doing well, right? Yeah, by the back. And it's and she's pouring a lot of alcohol. I'm like, oh. So we saw the packet of Chris is checking you out and then realized it was you.
Magic. Sorry, what you got?
I didn't even notice that. Good point. Chris, what are you trying to say? Yeah, then he's like, come on. I was like,
Eve.
I'm here.
Yeah, Chris, I'm moving you.
Why?
Huh? Because I want to.
Why? I'm not bad.
Can I put you next to Harrison, please?
Yeah, why? Why is he moving? I don't know. Wait, Chris. That's so funny. She knew my mom's name was Catherine. Like, he's like, come on.
Guys, I'm trying to take roll, but everybody's out of the seats.
Yeah. Well, already Catherine. You do?
Huh? Alright, I have taken 6 cell phones today. If I see them anywhere near being even out, whether you're on it or not, I'm going to snag it. I guarantee you that I'm taking somebody's phone today. Guarantee.
I'm waiting for Mr. Butler. Mr. Butler. Junior Butler.
Yes, props. All right, listen up. I set it to. Distracting everyone. They're all staring at you. You can't pay attention to me when they're staring at you. Okay. Right now you should have a piece of paper out and be going on number 12. I'm going to call on you guys in about 3 minutes. These answers are based on the reading from A section 1, which you were supposed to do after the test from last class. Sorry I haven't got your scan front all fully graded. So I'm doing that this morning. Most people got a D or F on the test, but you know, whatever. Yeah, you guys hang. You know, most of you guys in this period flunked the test. It's period 6, right? Yeah, most people got an F. It's out of 40, you're looking, you know, F. 19 out of 40, that's an F. 31 out of 40, that's a C. 7 out of 40, that's a negative, like that's, I can't even, that's like a Z. It's not even an F, it's like more like a XYZ. 20 out of 40, F. 32 out of 40, that is a B. 35, that's gonna be an A.
Who is that?
30, that'll be a B. Ish. 14, F. 17, F. 31, probably B minus C. 24, D. 29, C. 13, F. 23, barely on the cusp of F and D. Sign yourself. 22, F. 25, D. 11, because they forgot to bubble in the first 10. What? Make sure that we add those. 30. B minus C plus. 28, C. 37, yay, A. 19, F. 20, F. 27, D plus, C minus. 38, A. 15, F. 12, F. 21, F. 16, F. 22, F. 21, F. 30. No. I'm proving a point. 30. C plus B minus. 24, D. 19, back to the person that had 19, F. What's that? Oh, yeah, start sweet. Go with it. I don't know why everybody got such bad grades. When I gave you most of the questions and their answers on the PowerPoint. Do you guys realize that the iClicker questions are on your PowerPoint? Yeah, but that's not what I asked. I didn't ask that question. Do you guys realize that the PowerPoint questions are on your PowerPoint? Yes. And those are overwhelming the majority of the questions that I use on the test. So essentially, I'm giving you the questions and the answers to most of the test questions right before you take the test, and you still get an F. Am I missing something? Am I wrong? Do you think that I'm wrong by saying is that a false statement? Right? Because a lot of those questions that were on the iClicker were the ones that I put into the test. The first 10 are obviously the common assessment, which were the easiest questions on the face of the planet. If you guys didn't get at least 8 out of 10 on that, then I don't know, then you deserve to get the F that you got. Because it was super easy. Right? I don't know why you're getting such horrible grades on the tests. but I can assume as to some of the reasons why. Are you reading the books that the PDFs that I post that are due on Fridays? Ask yourself that. I never read that crap. I just copy and paste or not do it at all. Number one. Are you copying every single worksheet that we do in class from someone else right before it's due? Are you on your phone every second that I'm lecturing? Not paying attention to a damn thing that's coming out of my mouth. Do you ever actually go back and review your note packets? Do you ever open up your PowerPoints that I give you, okay, to look at them for a second time? The online quizzes that you take, you completely cheat off someone else who gives you the answers after they've taken it. If you do those 6 things, right, if the answer to those questions is, yeah, oh, wow, that sounds like me, that's why you have a crappy grade in my class. It's a simple formula. It's not Mr. Vieira's class is so hard. This test is so hard. We do so much work. Ugh. I wish I had Woodward. Ugh. Okay? It's not really that difficult if you just do some basic fundamental things. Do I have students that have A's? Crap, I got students that have like 109% in this class. Okay? And I have more than one. Are they just geniuses? No. They just do those 6 things that you're not doing. Okay? They're actually reading the book. They're going back over the notes. They're doing those questions on the margins as we go over the notes. They pay attention to when I lecture. They listen. They're not asleep. They're not on their phones. Right? They watch those videos that I post on in the updates maybe more than once because they know it's important. They actually try to answer the questions on the quizzes themselves, and they have 4 or 5 attempts, so by the time that they're done, they kind of understand what I'm trying to ask you. They're paying attention during the iClickers, and they go back and they review the answers to the iClicker questions directly before the test. They do all those things. That's why they get A's. It's very simple. Okay? There's no reason why you should have that many F's in one test. You know why? Because I've done this exact unit before millions of times, and I've never seen a class tank that test like that before. Never. I was shocked this morning when I ran your guys' scantrons. I've never seen anything like that. I've been teaching for 10 years. Never had that many F's on the cellular respiration and photosynthesis quiz. Well, whatever. That's on you. Anyway, now that I've completely crushed your soul, let me see how well you guys have done the warm-up. Who has not finished the warm-up? So, okay, about 3 minutes it'll take you. You can talk to your neighbor. I want you to talk to your neighbor. Discuss with he or she what the answers to those questions are.
Jordan, what the homologous
Yep. Hello. Hey. All right. Jesus Christ. You're a giant man.
I didn't like that at all. That was weird. Who was that?
Right, what is a chromosome? What is a chromosome?
A chromosome is a tightly coiled structure of DNA.
You are correct. What does it mean when I say the term homologous pair of chromosomes?
A homologous pair of chromosomes are two copies of each autosome.
Yes. Why is our DNA in a pair? What is that? Why did, I mean, I know when my shoes are in a pair, right? Because I need one for my left foot and one for my right foot. Chris? One from your mother and one from your father. So chromosome number 1 that I got from my dad matches up with chromosome number 1 that I got from my mom. They have the same homo genes, logos. They might not have the same version of those genes. They have the same gene. Somebody asked me if I look more like my dad than my mom, it's because I have more of my dad's DNA. No. You have 50% of your dad's DNA, 50% of your mom's DNA. Typically, if you look more like a parent, because the genes that control this part of your body, you may have, like, let's say you have your mother's shaped eyes, and because they were more dominant or whatever, and your nose shape is more like your mother's, even though you have your father's and everything else, whatever, that's, people tend to think that you look more like that parent where your eyes and nose are shaped. That's it. That was my only response to that. Yes?
So it's two copies of your DNA?
No. I'm going to... The copy of our DNA, when it's attached, is called the sister chromosome. We take our dad's chromosome number 1, we copy it. That's that X. Mom's chromosome number 1, copy it. That's that X. The two together, the two X's, they are a pair. A homologous pair. Dad's chromosome 1, mom's chromosome 1. Dad's chromosome 2, mom's chromosome 2. All the way to our 23 pairs. They are a pair, they are homologous, they have the same genes. How does a chromosome differ from a chromatin, which differs from?
It's right here though.
Oh, I thought you were absent. When you snuck in, then I marked you absent. I got marked you here.
No. I said she was part of
Miguel, do you know the difference between a chromosome and chromatin? Did you know the difference between a chromosome and chromatin? No? Yes, Sam.
Okay, so a chromosome is in cell division, and the other one, the DNA and the protein, are in non-dividing cells. That'd be right. Because
What?
non-dividing-
What's chromosome versus chromatin?
I know that one was in non-dividing cells, and
No.
that's what it
Okay, I see what you're saying. A chromosome is what is in a cell that's dividing, and chromatin is a cell that's not dividing.
is. Yeah.
Yeah, you don't see our DNA packaged up as a chromosome unless the cell is dividing. That is correct. You are correct in terms of when you will see these structures. What about in terms of their physical structure? Like, what do they look? How do they compare in terms of like their actual physical structure? You're right. Chromosomes only when a cell is dividing. Chromatin is when a cell is just normal. Yeah.
Chromatin is like extended DNA, while chromosomes are like condensed DNA.
Yes. So then, how does DNA itself compare to chromatin?
Chromatin or chromosome?
Chromatin.
Chromatin. Is chromatin part of DNA?
Chromatin is DNA, but it's wrapped around what?
Little proteins.
Little proteins. But it's still not as densely compact. Here's how I explain it. This thread right here, okay? This thread represents DNA, okay? We have 23 of these long molecules that I got from my dad. 23 long molecules I got from my mom, okay? This thread is DNA. We're super long. DNA is super long. Okay, this is one molecule of DNA, one chromosome. Got it? From here to here, that's a gene. From here to here, another gene, another gene, another gene, another gene. A gene is a piece of DNA that's going to have information that codes for a protein. That protein does something in my body. That's going to give me round eyes. That's going to give me curly hair. That's going to make me colorblind. That's an enzyme. That makes me ridiculously handsome. You know, whatever. Like, those are all the genes that you have, right? So this is our DNA. It's really long. We have 23 of these we got from our dad. We also got 23 that we got from our mom, and they're just all, you know, in our nucleus. Now, they're actually wrapped around these things called histones. So DNA, when it's wrapped around histones, it's called chromatin, right? So imagine this, but a little bit more packaged, right? It's a little bit more like packaged up like this, right? Here we go, like this. That is chromatin. Just wind it up a little bit more. Yeah, packaged DNA, but it's still really long. It's still long and thread-like. Now our cell's about to divide. The cell's about to divide, I need to make a copy of this, right? Okay, so I would make a copy of this, and then I would coil it up even more. So I am like going to really, really coil up my chromatin, which is wrapped around. Really, really, really gonna package it super tight, right? And then I have this thing right here. This thing that's super, super, super tight. That is a chromosome. But remember, guys, I had a copy of it, right? I had to make a copy of that. I buy these for this reason. I made a copy, an identical copy of Dad's chromosome number 1. It's an identical copy, and I attach it in the middle. Right? So, let's see if I can do this. Hold on. Now these things are like sausage-like. Right? They're like really, really, there we go. Here's a chromosome. Right there. Here's another chromosome. Right here. And they're attached in the middle at a centromere. That's a little X. Okay? That is a chromosome. Dad's number 1 and a copy of dad's number 1. Mom's is somewhere else, making a copy and attaching it to it. This happens during interphase, making the copies. It coils up during prophase. So when you see these little X's, right? When you see this, there we go. See that right there? See how that's a chromosome? It's really one chromosome and it's copied. There's a million different pictures that you can see. Here's another picture, right? Remember, it's DNA wrapped around histones, coiled up, and it's copied, attached. That is dad's and dad's copy. No matter what I do, no matter what I look at, how many millions of pictures, DNA coiled up, and it's copied, attached at the centromere. A million different pictures, represented by a million different artists, all showing the same thing. Once again, chromosome, copy, attached. Each little piece is a gene, right? Piece of DNA, piece of DNA. There are many genes on one chromosome because one chromosome is super long. I could have like 1,000 genes on chromosome number 1. Now this picture of all of my chromosomes right here is called a number 4. What is number 4? For your warm-up? That's a karyotype. A picture of all of your chromosomes. I could have a picture of a human cell, I could have a picture of a plant cell. Do they look exactly like that right next to their homologous pair in the cell? Of course not. This was taken and then arranged by a computer. They actually look more like this, right? This is the nucleus of a cell during my metaphase, right? But all lined up in the middle this way. Snapshot. I am a trained geneticist. My job is to make karyotypes. I then, using computer software, match up the chromosome pairs, the homologous pairs, right? Each one of these is called a sister chromatid. See how the sister chromatids are attached? That's one chromosome. That's another chromosome. That's another chromosome. I can match them up based on their size and band pattern. And when I match them up, right, then I can have what's called a karyotype. 1, 2, 3, 4, 5, 6, 7, 8, 22 pairs homologous, and that last pair is my sex chromosomes. If I have an X, which is kind of big, and a Y, am I a boy or girl? If I have 2 X's, I'm a girl. It's the presence of the Y chromosome that determines biological sex. You either have a Y or you don't have a Y. You can have 3 X's. You can have 1 X and no Y. You can have 2 Ys and 2 X's. There's a lot of combinations that you can have because it screws up when we have fertilization. There's a lot of things that screw up in meiosis. But at the end of the day, we determine sex by the presence or absence of the Y chromosome. Gender, right, whether you associate yourself in society as a male or female, is a sociology term. It's not a biological term. You can be born XY, male, that's your biological sex, and then have your gender be female because you associate yourself as a female. And you can have that legally changed. So your driver's license will say F, female. Not because your sex is female, because your gender is female. And so legally you're female. You can chop up, you can chop off your penis and do whatever you want and do gender reassignment surgery and go pee in the girls' bathroom for the rest of your life and nobody can do anything about it. And that's the difference. It's not called a Y chromosome because it's shaped like that. It's not shaped like a Y. It's shaped still like an X, but shorter. Okay? All right, is everybody done with this? Number 3, when do you see chromosomes in a cell? What is the cell going through? Yes? You're getting too specific. Yeah. Cell division. Whether it's going through mitosis or meiosis. If I walk into your house and all your stuff's packaged in boxes, what are you doing? Moving. You're moving. If I look at a cell and all of its DNA is coiled up and attached to a copy, so the DNA looks like a chromosome, that cell must be going through some sort of division, whether it's mitosis or meiosis. So there's a 6-minute video. He's going to give you an overview of mitosis. This is the best video I've ever come across on mitosis because it covers the entire cell cycle. It goes over the cell cycle. It talks about the stages of mitosis. It compares animal cytokinesis and plant cytokinesis. And it talks about all the checkpoints, which is how a cell regulates whether it's going to divide or not, which is the definition of cancer when the checkpoints get like ignored. 6 minutes. I posted a link to this video on your Schoology. So if you ever like, I don't know, get confused because you're not paying attention to me now, you always pull this video back up at home.
We rely on cell division. In eukaryotic, the adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into 2 identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleus.
Is that moms and dads?
No.
That's dads, Anda. Copy. What do we call these two things? Because they're identical, they're called what? No. That's not the homologous pair. That's dad. His pair is the mom chromosome. These are identical, not the same, they're identical. Chroma, sister, chromatids, with a D. This is chromatin. This is chromatin right here. That's chromatin. Wind it up even more condensed, chromosome. From least to most condensed. That's an online Schoology quiz question, by the way. You have to rank it in order. DNA is the loosest. Chromosome is the most tightly compacted. I have a picture of a chromosome and I ask you, what are these 2 things right here? They're called chromatids with a D. Sister chromatid, like twin sisters, yeah.
How could you tell if that was using
I don't, it could be mom, I have no idea. That's a great question, by the way. Yeah. No, it could be the mother's chromosome.
Chromosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into 3 distinct phases. During G1 or GAP1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or GAP2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as 2 sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach by proteins in their centromeres called kinetochores to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint, and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all three phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide.
All right. Take out your notes.
Me too, me too.
Yeah, push your back together. You guys scoot your table back to the movement. Everybody, you guys move back. You guys move back. You guys move back.
Can I take the test during
Huh?
Can I take the test during your tomorrow?
Sure.
Thank you.
Okay, so it's about cell cycle. Cell cycle is the life cycle of a cell in between divisions. That's 4 parts: G1, S, G2, and then the M phase. And we're going to learn about what goes on in all those steps. There are lots of pictures in this, so we're going to learn through videos and pictures, because this is a process that has a start, middle, and end to it. Okay? It's like a dance, right? It's the beginning of the dance, the middle of the dance, the end of the dance. And every part of the dance, some parts last longer than others. Okay? And so this is what we're learning about how cells divide. The video showed you what happened in each step. Now we're going to talk about not only what happens in each step, but why a cell has to divide to begin with. Originally, we thought that cells just had two phases. When it was dividing and when it wasn't. When it wasn't dividing, we call that interphase. Inter, like in between divisions. The M phase, what do you think the M stands for? Mitosis, yeah. Well, that's kind of what we associate it with now. Now, we have a little bit better tools now. We actually saw that interphase is broken down into 3 subphases, G1, S, and G2. Which is longer, interphase or M phase? Yeah, clearly. Look at the size of the arrow. So what do you think a cell spends most of its life in? Interphase. I like to call it the just chilling. It's just doing its job. Right now, 99% of your cells are in interphase. G1 of interphase. G1. It's making proteins, doing its job. At some point in time, each cell, different times, it decides it needs to divide. There are reasons cells must divide. They get too large, they get damaged, you're growing, whatever. When they divide, they go into S and G2 and then M phase. And we're going to talk about what happens in all this. So this is the just chillin' phase. G1. G stands for gap. You can think of it as grow. Grow. Cells growing, doing its job, making organelles, making proteins. Nervous cells are firing electrons or electrical signals. Muscle cells are contracting. Bone cells are producing the hard part of the bone. Immune cells are making antibodies. You know, your cells are doing what they're supposed to be doing. Now, at some point, that cell might decide, you know, it's time to make a copy of myself. And then it goes into the next phase. The S stands for synthesis. Now, for those of you who are pretty good at English and understand what synthesize means, to synthesize something means you're doing what? You're making something. You're putting it together. Well, what am I synthesizing in the S phase of interphase? Nope, not a new cell, more specific. More DNA. This is where I make my copy. You guys remember when I had the thread and I made a copy and I attached it together? All that happens here. If I'm going to make a copy of my classroom here, I need to make actual human clones of all of us. Right? I need to make a clone of you. If I want to make two period 6 Mr. B classrooms, I need to make a clone of all of us. And then I'd attach that clone to our hip. That's what happens in S phase. We make a clone of the DNA and we attach it at the centromere. Okay? Now after the S phase, we want to make sure that all the DNA is copied, we have enough ribosomes, we have enough mitochondria, everything is checked and ready to divide. That's the G2, that's what happens in G2. We are preparing for final split. Once G2 is finished, now we're going to start the division of all that DNA, and that's called mitosis. This is a critical checkpoint, right? We're going to wait to make sure everything, all the DNA has been copied and everything's ready, it's all attached. Then we enter into the M phase, which is mitosis. If you look at this diagram, mitosis, the M phase actually has 2 parts. It has mitosis and cytokinesis, which are not the same thing. Mitosis is the division of the nucleus and all the DNA. See, I can divide the nucleus and still be in one cell. Cytokinesis is actually splitting the cell cytoplasm. And that's what happens towards the end of mitosis. And it's the final thing that occurs. Plants and animals go through cytokinesis differently. And that was shown in the video. Animals just pinch in and form a cleavage furrow. Plants can't pinch in because they have a cell wall, so they just build a wall in the middle, and that's called the cell plate. Those 3 words hard to write? Do bacteria cells go through mitosis? Since mitosis by definition is the division of the nucleus, do you see a nucleus here? No. Bacteria cells do not have a nucleus. They don't have chromosomes. They just have one circular DNA. Prokaryotic cells don't go through mitosis. They go through a division process called binary fission. It's different. It's quicker, it's faster, it's easy. You just make a copy of this loopy DNA, attach it over here, boop, and we have 2 cells. Prokaryotic cells, bacteria, they can copy themselves in like a half an hour. Our normal cells, eukaryotic cells, by the way, this cell should be super small. Is this to scale? No, not even kind of. Bacteria cells are tiny. These cells take hours to divide. Is every cell always dividing every 12 hours? No. We're not always dividing our cells. In fact, some cells never divide. Our skin cells have to make new copies every 2 weeks or so because we're always rubbing off layers of skin. Imagine if our skin cells never made new copies. We would damage the top layer of our cells and we would never replace them. We'd be like walking skeletons that our muscles would be exposed. We need to constantly make new skin cells. It's the same with the inside of our intestines because we're always eating food and it's scratching up against the inside of those walls. Liver cells get replaced every 2 years because sometimes they get damaged from all the poisons that it has to filter out. From the video, what 2 types of cells never divide? Yeah.
Brain cells.
That's one type, yeah, neurons, right? And what else? Muscle cells. You're probably thinking, hold on, if muscle cells don't divide, then how do you get stronger when you lift weights? Isn't that more and more cells? What happens is the cells get bigger when you work out. You rip apart the cells and all the proteins inside of that cell. And when they repair that cell, they add more proteins. Muscle cells are like cylinders. And in the middle are the filaments that slide across each other and cause that cell to contract. When you work out, you destroy those cylinders. And when they build those filaments back together that are made up of proteins, they build more. So your muscle cell gets bigger, thicker. So that's where you build your muscles up. But you don't have muscle cells divide. So if you're born with 7 trillion muscle cells in a muscle, that's how many you're going to always have, but they can become bigger. Takes about 12 hours. That's completely average. That's not, guaranteed. When we're growing as a little tiny egg in our mom's uterus, an embryo, our cells are dividing very fast. You get to be from 0 when you're pregnant to 10 months later, 40 weeks later. It's not 9 months. It's 10 months. How many weeks in a month? 4. How many weeks are you pregnant? 40. How many weeks in a month? How many weeks are you pregnant? 40. You're pregnant for 40 weeks. Are you 9 months pregnant?
9 months.
You're 10 months pregnant.
So then why is
Leap year.
Not every month. Not every month is 4 weeks, some. You're pregnant for 40 times 7, right? 280 some days. Which is about 9 actual calendar months. They don't tell you you're 3 months pregnant at the doctor. They go by weeks. You're 27 weeks. You're 32 weeks. When you get to be 40 weeks, that's your due date, right? But if you think about it, right, technically, if you think of a week and 4 weeks is a month, and 40 weeks, that's 10 months, really. That you're pregnant. 10 months. Not 9. 10.
so then why do people?
They've been lying to you.
I mean, it makes sense. I just never heard it.
Yeah, well, if you actually look at 9 calendar months, it's closer to 40 weeks than 36. Does that make sense?
Yeah.
Remember this lab? Raise your hand if you remember this lab. What color were the cubes? They were purple. We put them in a cup of acid, and they turned what? The reason why I did that lab was for you to realize that as cells get bigger, there is a problem, a mathematical problem. What grows faster, the volume or the surface area? The volume. And the surface area can't keep up. So this cell sucks up nutrients way better than this cell. Does that make sense? So if I had this cell, I need to chop it into tiny pieces because its ability to absorb nutrients is not very good because the surface area is not catching up to the volume. And the surface area, which is the outside of the cell, refers to the plasma membrane. And so if a cell gets too big, it cannot absorb nutrients or get rid of wastes fast enough. So how does a cell solve this problem as it's getting too big? What would a cell do? It's getting too big and it can't suck up enough nutrients or get rid of enough waste. What would be a solution to this problem? To divide, to split in half into two smaller cells. Yes? Part B?
No, the upper limit due
For C, it goes 1.
Right there?
Yeah.
Snuck it in there, silent. Like a ninja. Got it?
Yeah.
So this is a reason why cells need to divide, because they cannot absorb nutrients or get rid of wastes fast enough through their membrane, because as a cell grows in volume, the surface area cannot keep up. If we're going to divide, in order for that cell to be equal to the copied cell to be equal to the parent cell, this has got to be coordinated. It's not some random process. It's highly coordinated, like a dance, a choreographed dance. The chromosomes always do the same thing. Mitosis is that process, highly coordinated. I expect you to somewhat know that process. Ah, what is this class?
Carrier.
Boy or girl?
Boy.
How do you know it's a boy?
Because the accent.
See this picture right here? This is an actual electron microscope picture. This is some dude's drawing of this. Does that make sense? That's a cartoon drawing of this. Now, here's where students get confused. Is this little X one of these or both of these together? It's one. If I were to zoom in on this guy, right? I mean, he's curved, but if I were to straighten him up, chromosomes aren't like stiff X's, right? I mean, look at the thread that I handed her. You can move it around, right? So sometimes they're curved. They're not always like a perfect X. If I were to take this guy and zoom in on him, he would look like this. So technically, there's actually 1, 2, 3, 4 sister chromatids there. A better picture, right? Would be like this. Does that make sense? That's zooming in a little bit more. Do you see how there's 1, 2, 3, 4 sister chromatids? What do we call these two things together? Homologous pair. That could be dads and moms. I don't know, maybe that's dads, maybe that's moms. Can't really tell by looking at them.
Isn't that like the most normal?
Huh?
Isn't that like the...
This is a normal human male karyotype. Could I get a karyotype of a monkey? Could I get a karyotype of a plant? Sure. They're probably not going to have 23 pairs. What's wrong with this person?
They're missing
They're not connected.
Come on, I hired you. You're a geneticist.
I'm sorry, what's the joke?
They're
They're missing, there's a gap between 50 and 60.
No, guys. The chromosomes are like that. We take a picture, and then through a computer, the person matches them up.
Oh.
This is actually the picture that the person at the clinic looks at on the computer. This is what it looks like. This is what the chromosomes are kind of spread out in your cell in the middle of some sort of stage. If it's not in like perfect alignment. So they're then going to match them up. They're then going to match them up like this, right? What's wrong with this person? Yes.
The X and the Y.
X and the Y? No, guys, it's a computer image. I can put them close. It doesn't matter.
What about 21?
What? Problem.
That was it.
Is he a mutant? This is Wolverine right here. Yes. This person can move objects with their mind. No. This person has Down syndrome.
Yeah.
This is Down syndrome. No one wants Down syndrome. Well, this is only like a picture. I don't know why. If I'm a doctor, maybe. And I didn't even pay attention to that. I gotta visit this page and see what's going on. Who the hell is buying this crap? What? You out your goddamn mind. Is it like framed? Who's buying this shit? Are you kidding me?
I love how it's more expensive for the color.
All right, well,
Wait, what's wrong with them? They're missing.
yeah. Thank you.
What you missed? What's you missed? What did you miss?
Okay, so that.
Hey, you want that for Christmas?
Okay, everybody see how this is Down syndrome? Pair. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12,
It looks like
13, 14, 15, 16, 17, 18, 19, 20, 21. Look, 21 has 3.
No, it's right there, that middle one.
Not the middle.
It's right here.
It's just there.
Looks like they're dancing on the cross.
I don't know. I don't know. And somebody asked me, why is it all hairy? I said, well, do you see how my little threads look like when I coil them up? You know what I'm saying? Understand what a chromosome is. A chromosome is a long, thin molecule condensed. So that's that long, thin molecule. It's not perfectly smooth. If I take a thread and I coil it up, is it going to be smooth on the edges?
No.
It's this. This is what's going on. Unless you give it to one of your students and then they try to unthread. Your finger is going to fall off. You are cutting off the blood supply to the tip of your finger.
I forgot to turn something in yesterday.
Yeah, not a problem.
Hey.
Sorry.
Whoa.
I posted an online Schoology quiz. That's how I roll.
You're so nice.
You cut into my Black Ops time because my mom has your texting.
Nope. Hell no. You think I want your random text back? Like at 1 o'clock in the morning?
Oh, we should get like a. We should start like
I had a student send me a picture of like a bottle of vodka and then like, you want to know that you're getting drunk on a Tuesday? 1 o'clock in the morning? Dumbass. Yo, B, can I send you pictures? I don't want pictures from you. Yeah, you cannot send me pictures.
Ooh.
Thank God.
Someone's
That's
naughty.
I'm like, what part of sending me a picture of you getting drunk What part of your well,
Only females have the advantage.
no, I was like, look, it said Mr. V on it.
I mean, if it was
I only did this once, I'll never do this again. I gave an extra credit assignment. When the Niners went to the Super Bowl, what, 3 years ago or something like that? 2 years ago. 2 years ago? That's 2 years ago. When they went to the Super Bowl, I said I would give any of my students extra credit if they took a picture of themselves in Niners gear rooting for the Niners. But then I was getting like all these like pictures of girls and like Niners, so I'm like, whoa! My wife's like, what the hell is that? Should have been a little bit more clear how I wanted these pictures. Not good.
Girls are gonna send you anything correct.
I was like, yeah, send me a picture of you like being a fan of the Niners. Not like send me a picture of you all scantily clad in like a Niners tank top in the bathroom doing one of them selfies or whatever. Like, no. Not what I intended. So no, I will not offer that extra credit assignment ever again.
Oh, come on, V.
What if they win the Super Bowl?
I did have a Raiders fan, student of mine who was a Raiders fan, and he sent me a picture of him flipping me off. Yeah, like his dad was a Niners fan, so he had a Niners shirt, and just like,
Here's your extra credit, B.
douchebag. Why you're a Raiders fan? Alright, there's going to be a test question on that online quiz that has a picture. It has a picture of a chromosome and like an arrow pointing to here, and 2 arrows pointing, you know, like these 2 things are labeled, and you have to label it, is it looking at the chromatids or the centromere?
I keep on thinking you say chromatid.
No, chromatid with a D. Not chromatids, dude, come on.
Hey, Malia said it, not me.
You know that Phineas and Ferb episode where they talk about the aglets?
No, I did not.
I had no idea of what this show was or that this episode, but a telomere, everybody does. At the end of our DNA are these things called telomeres. At the tips of our DNA are things called telomeres, and they kind of act like aglets in the sense that they keep the chromosome from falling apart. Kind of like those plastic things like your shoelaces keep the shoelaces from unraveling, right? Yeah. So telomeres act very much like those aglets. And as we get older, these telomeres start to break down. And the breaking down of our telomeres is one of the reasons why we age and die. Because it kind of like, our DNA just starts to kind of fall apart. And when that happens, then the cell dies, you know. And so if we can keep our telomeres intact longer, we can live longer and be healthier. Some of the things that cause your telomeres to break down: eating a bunch of crappy foods, smoking cigarettes, exposure to UV radiation. So if you've led a pretty harsh life, your telomeres are getting shorter. You're going to die quicker. They are like aglets. At the tips at the end, kind of like at the end of a shoelace, at the end of DNA, at the end of a DNA molecule, at the end of a chromosome, right?
Yeah, what does this have to do with my...
Oh my God, shut up.
Let's just
We
move
were
on.
just comparing.
Bad analogy. Bad analogy.
That's sick.
It's a perfect analogy, but bad.
I can say something back, but I'm not going to.
It's a shh. You don't know what or when? It's usually like the end of July, the beginning of January. The end of January. Something that's shady. Hey, it's been a long day. Shut up. Fired. Alright, so we're going to stop. Alright, so we're going to go over all the stages of cell division, starting with interphase before the cell divides. And then we're going to go through mitosis, where the cell's nucleus divides into 2 genetically identical nuclei, right? We're going to take our DNA, we're going to copy it in interphase. Look, this is 2 chromosomes, right? Chromosome number 1, dad, chromosome number 1, mom, chromosome number 2, dad, chromosome number 2, mom, or vice versa. That doesn't work out. look, we've copied it. See how it's now, look, can we kind of see how it's copied here? Now we have the little X's. This right here, interface. Then we're gonna line them up in the middle and separate them. All this right here, mitosis. Splitting the cell in half, cytokinesis. So, mitosis, the division of the nucleus, has 4 parts. PMET, 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 PMET. Prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. In prophase, there's a lot of stuff going on. Nuclear membrane breaks down. DNA coils up into chromosomes. Centrioles migrate to opposite ends. I have no idea what that is. All of this stuff happens in prophase. You are required to know. You guys are required to know what happens in all of the stages. Or if I show you a picture of a cell, you can tell me what phase it's in. Okay? After prophase, we got to line up all those chromosomes in the middle of the cell. That's metaphase. Middle. Meta means middle. Pretty obvious. They're attached to these spindle fibers. Spindle fibers are these microtubules made of proteins that spawn from these structures right here. What is that structure right here called? Centriole, I heard it. Centrioles are packages of microtubules, and the microtubules, they build themselves into a spindle fiber and attach themselves to the middle of the chromosome at the centromere on a protein called a kinetochore. And then they line them all up in the middle. And now what happens is each sister chromatid is going to walk along. These guys over here, and now we're going to separate. Imagine you and your twin attached by the hip. Right? I made a copy of all of you. You and your twin attached by the hip. We all line up in a single file line. And I asked your twin to go that way, because he's like this big old spindle fiber attached to him, and the spindle fiber attached to me. And then we just start, I move to the left, and my evil twin moves to the right. And so that next part where we separate the chromatids, we separate the copies, anaphase. Yes?
Wait, in 7th grade we did like this, book, I don't know how you say it, like
Flip book.
stop thing, like when you're recording it and take a picture and then...
Yes.
Yeah, we did that when we made those out of candy. It was like licorice in the end.
That's fantastic. So this should be easy. I have a project called the Mitosis Project, where you either make a poem, a flip book, or a music video.
Why do you make it like you don't want to do any of that?
Huh?
Like, none of them are.
I may not assign this.
Could it be extra grade?
Huh?
Extra grade?
No, I'm either going to do it as an assignment or I'm not doing it at all.
The second one's better.
Standard 7th grade.
I have a lab where we do like Oreo cookies and we put those little sprinkles, you know? And the sprinkles are the chromosomes. And you open up the Oreo cookie and it's like the nucleus, right?
You didn't do that last year.
I have a lab. I didn't do it because we ran out of time. You guys are going to do a lab where you have to select things that are in each stage, but it's on your iPad. The iPad cards come in next class. You're going to do a little mini lab on your iPad.
I have
It's pretty easy.
Did you go to high school?
no, some people return it.
I don't
High school in a different part of the state. I'm not from LA. I'm not from Southern California. Can't you tell that I'm not from Southern California?
Yeah, it's
Isn't it pretty obvious?
Where are you from?
The farmland.
I'm from where fat people and meth come from.
Where are you from?
Carfax and cheese.
Where are you from? Marion?
Very, very close.
Really?
Right south of Stockton, Vanessa. I'm from the ghetto. All right. After anaphase is telophase. I told you I'd take one. I told you. Oh, Amber. It's okay. You're the 7th cell phone I've taken today.
Yeah, I know.
So, now that we have 2 nuclei in the cell, right? Prophase, we prepare. Metaphase, we line up in the middle. Anaphase, we pull apart. Telophase, we have two nuclei, one cell. Cytokinesis, we cut the cell, cut this, I don't know. Separation of the cytoplasm. I have no idea. I don't. Go. Because it starts with the letter T. What are you talking about? I stands for interphase. P stands for prophase. M.
Anaphase.
Telophase or telophase. Guys, I just want you to respond what I'm asking you to respond. I don't need the extra 75,000 comments. C stands for cytokinesis. Guys, that's it. Just do what I ask and cut out the extra part. PMAT. This is mitosis. How do I remember the order? Interphase is start. That's where the DNA is copied and the cell's just chilling. Then a cell decides to divide. Now we need to divide the nucleus. We go PMAT. Prophase, metaphase, anaphase, telophase. And then at the very end, we separate the cytoplasm. We split the cell in half. That's cytokinesis. IP over MAT, see? IP on MAT, see? That's one way that somebody tried to teach me, but I didn't like that. So I use a mnemonic device. I see your hand. It's going to get tired. So just wait. Hold it at the end. Thank you. A mnemonic device is like, please excuse dear Aunt Sally. I used to have an assignment where I actually had the students create a mnemonic device to understand the order of events in the cell cycle. Like, I play Madden at Tommy's Casa or something. Or I punch monkeys at the church. Right? But what I used to tell my kids is I gave them my mnemonic device, which is I pimp moms at the corner. Now, I got that from another teacher. She was like 50 years old, and I'm sitting there as a student teacher, and she's like, yeah, so we're going to do a mnemonic device, and let me give you an example of one. I pimp moms at the corner, and I'm like, what? What? Did that just happen? So I teach that, right? I say that. I pimp moms. I, interphase, pimp, prophase, moms, metaphase, at, anaphase, the, telophase, corner, cytokinesis. So I used to say that, and then I would say, okay, guys, make your own mnemonic device. And so everybody would try to, like, one-up me. And so I would get these inappropriate ones back. You know what I'm saying? Like, all the boys were like, oh, check this out. I'm like, I got it. So yeah, stupid stuff. Like, I pinch my ass tightly, cuz. So I stopped doing that. So instead of doing, I stopped. Because there's no point. What's the purpose of doing a mnemonic device? To memorize the order of something, right? Are you ever going to forget I pimp moms at the corner? Never. When you guys are in the middle of the test and it asks you the order of the stages, and you have multiple choice questions, what's going to pop into your head? I push my ass out the whatever works. Now, I was at the mall 5 years ago, and one of my students that had graduated maybe 3 years prior to that, I'm in the middle of a busy mall. It's like Christmas time. I'm with my wife, and I might have had my daughter at the time. Busy food court. All of a sudden, I hear screaming from across the food court, "Yo, Mr. V! I pimp moms at the corner!" The wife would Hope it's not my mom. Like, I don't even know if you remembered what that stood for, you know? You had me for a whole year, and all you took away from that 5 years later, I put mom to the corner. I don't even know what that means. So, no need for the mnemonic device this time. You will never forget the order.
No, now we have like here. Like that's good.
I put my ass silently because you gotta do that. It's important. That's the cytokinesis, right? Some of them are just dumb. First of all, you got the letter P in there. People got a little too. I had to crumple up a lot of pieces of paper.
N, A, and C.
Yes. Oh, see? Really? Come on.
Dude, I'm just saying.
All right. Anyway, moving on. So here's what I want you guys to do. Everybody took this piece of paper, right? Check it out. On the back, it wants you to draw the phases of the cell cycle. Sometimes I have students draw that picture over there. No, I don't want you to do that. I don't want you to draw the arrow thing. I want you to draw the phases of the cell cycle. Draw a cell in I, P, M, A, T, and C. Draw a cell that's in interphase and label it. For example, this is a cell in interphase. In interphase, the DNA is uncoiled, chromatin. It's making the copy of itself. You got your centrioles on this side. You got your nuclear membrane still intact. You got your cell membrane on the outside. Now, that would be one drawing. I drew it and I labeled it. What is after interphase? Prophase. You know that.
Yeah, I don't know.
Pin, punch, right? Pinch. I still got my cell membrane. My centrioles are now moving, opposite ends of the pole. My nuclear membrane I drew as a dotted line because it's breaking down. My chromosomes are now not in chromatin, but they're coiled up now, so I can see them as little X's. I use two colors to represent mom and dad. I have two pairs here. I didn't draw 23 pairs of chromosomes. I'm not going to draw 46 chromosomes. Let's just say that this is some fake organism that has two sets of chromosomes, two homologous pairs. These two go together because they're the same size, and these two go together because they're the same size. Chromosome pair number 1, chromosome pair number 2. These are homologous pairs, mom, dad, mom, dad. Okay? Now that is prophase. I labeled the centrioles. I even labeled these two things as being sister chromatids. This whole thing is a chromosome. What would my next cell be? Metaphase. So what would those 4 X's look like?
They'd be
in the middle in a single file line. And then anaphase, I pull apart the X's and I no longer have X's anymore, I have singles. I need you to show me 4 chromosomes, 2 pairs in all your drawings. It would be smart to use different colors for the moms and the dads, right? So like get a red, blue, or whatever. All you need is 2 different colors. Because I'll be looking at that, making sure I will subtract it points if you don't show the spindle fibers attached to the chromosomes in the right way. This is a great drawing. What is it missing?
Color.
All the what?
all the
Labels. Nothing's labeled there. Are those the right stages? Yeah. In fact, it actually breaks down prophase into 2 parts. Because prophase is so long, it has 2 parts. It actually is called prophase and prometaphase, sometimes called early prophase, late prophase. You guys don't have to draw both, just pick one. This is like in the beginning of prophase, and this is what it'll look like towards the end, because there's a lot going on there. So where am I going to get the definitions or the events that occur in each stage? On my notes. Or in the book. In the book. Where am I going to get pictures? In the book. If you have an iPad, you can Google it. If you have your phone out, it better be flat on your desk. And the only thing as I walk by when I look at that screen better be a picture of some cells. Because if it's anything else, if you got to go sweeping towards your cell phone to go close it or change it to something, I'll snag it. When you're done with this, it goes into the bin. If you get done early, which you may, this little matching, can you match the picture with what is going on? And we're going to go over this as a class together. is draw lines.
So this is definition.
This absolutely, yeah, definitions on this side, drawings on the back. This goes in the bin when it's done. Any questions? Yeah. What if you what? I can't do.
Like art. It's like just
Oh my God, you can copy circles and lines. Circles and lines, guys. It's circles and lines.
Could be the bulky
Do it fine. I got colors over there. Got it? Anybody need one of these?
What do you say to a getaway? Getaway.
Stop.
It's like. Because I don't watch soccer. Soccer is You look like someone that can almost correct. Spinning brushes. Advertisement on YouTube. Like magnets for weirdos. Talking about moms every time. No. It's like Excel. Your name is on Excel. See, even Joe Martin knows. I did this in the house. I bet you know what you're doing. Don't like that. How do you get money? That's the one to do. What did it say? Is it complicated? She looked like the devil all the time. She be like this. I hate I used to listen more. She got broke, so. Oh, damn.
I know. But it's because she didn't take it. She didn't take it. No, he like makes them make it like the last week of one. They have chromosomes what? Was it for algebra? I lived 2 blocks Around each set.
Yes, sir. Yes, sir. Can you just color which chromosomes are dad and mom? Yeah, but like I guess when I'm show me that
When it says label, guys, I know you're probably meaning just label the stage. Label some of the structures, some of the important structures. You don't have to do all of them, but some of them. Hey, we got
To the two new cells. Chris, we missed you. This is sad. It's really dull over here.
You're there. Are you sitting here now?
That's too far. I really can't do that.
Now label it. Yeah. Makes my app easier. Go to page 133. Yeah. 133.
Is it labeled too? Wait, 133?
Yeah. I just go. I need the one with the owl.
I have them all now.
Yeah, but this is a bad owl.
YouTube videos of them. Yeah. I'm sorry.
I hope.
Label.
That's really good.
Label those
I
structures.
don't even get it though, just do it regular.
The back of it's so thick.
You've never seen.
On the what? Equators, like the equator. The equator.
I got it. I got it. No.
Is that second circle the prophase?
Yeah.
Okay.
Let's talk about this.
Hey, Spirit,
Tuned out. What do you need? So I want one gone? Did it even turn on? Oh, God, that's going to be an interesting video. Today was an interesting one. Colorful electric.
Oh my gosh.
Yeah.
Actually, I want
I just made it all wrong.
You know what's really chill?